Welcome to area study 1.3, which means that we're in the booklet that says determining relatedness between species. And we're going to go through, there are a whole bunch of stuff that's cut for 2020, so make sure you check out sector as to which pages exactly are cut. On that very first page, you could cross off the whole bit that talks about DNA hybridization, and you could talk about the whole bit that you could cross off the bit about molecular DNA or mtDNA, and then you can cross off all the bit on master genes. So there's really only two parts to this area of study this year, and the first part that we're going to talk about is this thing called molecular homology. So just thinking about these words to begin with, molecular meaning these are something to do with a biomolecule. So what are the biomolecules that we need to know in this course? So we don't need to know lipids, we don't need to know carbohydrates. So what are the only two that we have left? We have proteins and we have, nucleic acids. So these are the two things that we're looking at here, the two molecules. Homology, the study of the sameness. So homo meaning same, ology meaning study of. So this is the study of similar molecules. And the only two molecules we do in this course are DNA or nucleic acids, but in this case, we're going to look at DNA and proteins. So to begin with, I want you to ask yourself a few questions. What is a protein? What is a protein made of? How many amino acids there are? What happens if you change the order of amino acids? Now, we have just focused on this, particularly with the development of when we were going through mutations. So you should be pretty familiar with this and what can cause that change in amino acids. Hopefully, you're therefore saying mutation. So let's come and have a look. Nowadays, instead of looking at just structures, so we talked about the similarity of structures when we talked about fossils, looking at the fossil record, looking at developmental biology, looking at comparative morphology like analogous and homologous structures. So they were the three ways that we did last little subgroup to compare species. But there are issues with those. So we're going to look at how can we molecularly determine the similarity between two species. So the first way that we're going to look at is comparing amino acids in proteins. The next one is comparing DNA. So what you can do with this is you can look at similar proteins that exist across different species and examine their primary structure. So we're looking at, we're looking at the primary structure, which is, what is the primary structure? That is the order of amino acids. And what this graphic is showing us is it's showing us how many amino acids differ from a human hemoglobin chain. So this is a hemoglobin comparison. So you pick in one protein. So we pick one protein and we're going to compare how many similarities, or in this case, how many differences there are between the primary structure of human hemoglobin versus gorilla, rhesus monkey, mouse, chicken, and frog. So all of those species all have hemoglobin protein. But when you look at it, you can see that the gorilla only has one difference in its amino acid chain. The chicken has 45 differences in that amino acid chain that codes for hemoglobin, or that becomes hemoglobin, and a frog has 67. So I think it's pretty logical to say the more similar your amino acid sequence, the more closely related the two species are. So what we do is we count up the differences in similarity, so the number of similarities, or in particular, usually the number of differences between two species. The less differences, the more similar. So they are, so therefore they are more closely related. And this makes sense when we think about it here. So here is a pretty big confusing diagram. I might go through it with you again in class. We have 6 species that come down the side here. And then this table here is just mapping out the number of similarities between them, but most importantly, it's mapping out the number of differences. So what I can do is I can have a look at species number 1 and I can read across. So here's the raw data on the left is the raw data. And then on the right is kind of a little summary table. So you can see here, species 1 has 2 differences between species 2 and 3 and species 5. and species 4 and 6. So what I would say is that species 1 is more closely related to species 4 and species 6. That's it. That's what I'm looking for here. So let's have a look at, say, species number 3. Okay, so species number 3, when we compare the number of differences that that's the number of similarities. I've got to read down. I've read that table a bit incorrectly, so I'm going to read this time. I'm just going to clear these pen markings and let's read down. So that was the number of similarities. See, I'm not reading properly. So the number of differences goes down here. So you can see here is species number 6. And as we read down, you can see that they are most closely related to species number 1 because they have the least number of differences. And they are the least closely related to species 2 and 3 because they have 3 differences in their amino acid chain. So it's about looking at your comparison. And we're going to go through some questions in class to make this obvious. Now, this table here, which is pretty much on the first part of page 2, can you write this, that it's not on the study design, these different types of mutations and different types of comparisons. However, I like to just really quickly go through it. And this is the fact that we were talking about different comparisons and different things that cannot change the protein very much when we did our mutations prac. So when we look at a conservative substitution, so these are talking about types of mutations. So if you get what we call a conservative substitution, it means that the amino acid changes. So this is a missense mutation, but it's a very similar amino acid, and so there's no change to the protein. You could also get a semi-conservative substitution where you replace it with something that has a similar shape, but there's a possible change in your protein structure. So essentially, not all amino acid changes are created equal, is all this table is saying. If you get a non-conservative substitution, this is where you get major changes in your protein structure and function. But I really want you to not focus on having to remember this. This is simply just a little bit of a highlight that I'm going through to help you understand that sometimes the amino acid can change and it can make a big difference, and sometimes the amino acid can change and it doesn't make much of a difference at all. Here's a nicer table to have a look at compared to that previous one. There was a bit too much going on. This is more likely what you're going to get. So this is cytochrome C. Cytochrome C is a protein present in the electron transport chain. It's also very important for apoptosis. So we've talked about cytochrome C before. And so it's present in all of these organisms. So what that means is the human has 1 difference in amino acid or the rhesus monkey has 1 difference compared to the human, whereas the yeast has got 51 differences. So if I said to you, which species is most closely related to the horse, let's have a look. So if we had a look at the horse, I would look down and I would say this is pretty close. It's only got 1 difference here. If I look across for the other 2 here, it's got 11, it's got 12. And so I have to look across both there to see what my similarities and differences are. But the horse is most closely related to the donkey. If I was to ask you what is the most closely related animal to the sheep? So let's try the sheep. So what's the most closely related animal? You can see that the sheep to itself doesn't really count. You can see that there are 2 differences between the sheep and the donkey and 3 differences between the sheep and the dog. So this means the sheep is actually more closely related to the donkey than it is to the dog. Bit crazy, isn't it? So sheep are closer to donkeys than they are to dogs when we look at their cytochrome C comparison. Now this little table here that we're going to go through, this is technically showing you mutations in DNA. So now we've gone back to comparing, actually I might do this one first and we'll come back to that in a second. So this is the comparison of DNA-based sequences, is the other way of looking at things. So you can either look at how many differences there are in amino acids, or you can look at how many differences there are in terms of just the DNA. So how many mutations have accumulated. So mutations accumulate randomly over time. But the more differences you get, the less, so over time, more and more differences accumulate. This means that you are less related. Yeah? So the more differences you get, the less related you are. It's the same as amino acids. It also means the converse, which means the more similar your DNA, the more closely related the species. Now, they've taken this bit off the course this year. This mutation accumulation can give us some kind of idea of what we call a molecular clock. So we know on average the mutation rate of a particular area of DNA. And so what you can do is you can say, okay, I know on average that this organism mutates once every 25 million years. Yeah? And so therefore at 25 million years, I should see there are actually two differences in the DNA because there's two branches that have come off and done that. And that's how I can actually use that and go, okay, there's two differences between these two species. I know that this section mutates at a rate of 25 million years ago. So, sorry, one mutation on average every 25 million years. So I can determine that they split from a common ancestor 25 million years ago. So yes, they've technically taken the molecular clock off of the unit design, but you will, the study design, you will see questions like this on papers. I'll just give you one more example just to highlight it again. So you can see here that modern lineages, there are 4 mutations different. There are 4 mutations different. So on average, yeah, we go back, and if I said 1 mutation every 25 million years, and they have 4 mutations, what does this mean? This means that they must have had a common ancestor 4 times 25, 100 million years ago. All right, so hopefully that helps with your understanding about the fact that we can use DNA or differences in amino acids in terms of our primary structure to determine how related two species are. In both cases, the more differences, the less related they are to each other.